Today we're going to talk about how to develop a scientific hypothesis. Now, before we get into this topic, I just want to be clear that this is a skill that takes a lot of practice. And while we're going to go through some basic overview and some examples today, it really is a constant process of doing this over and over and over again, sometimes for years, to figure out how to develop a really good, refined, and testable hypothesis. So to start, I really want to go back to the basic concepts we learned at the very beginning. And these are the basic things that are background to GBM research and what kinds of questions come up in GBM research. Because to develop a good hypothesis and a good question, it's really important to understand the basic background that is out there on the disease of interest, and also to understand what resources your lab has or your school has that might be available to you in order to execute a project. So let's start by reviewing this diagram. So by now, this diagram should be fairly familiar. This is our GBM standard of care overview. So when someone comes in with what might potentially be GBM, usually they're going to come in with, for example, headaches. They may say they're having balance problems. They may have localized sensory changes or motor changes. They're basically having some sort of host of neurological symptoms that is new, that makes their doctor think that maybe this person has a tumor and I should look for a tumor. So the way they look for a tumor is typically with an MRI. So you will get an MRI on this patient, and in some subset of patients, this MRI confirms that they have GBM. Now, I want to be clear that just because you have headaches or balance issues or neurological symptoms does not necessarily mean that you have GBM. It is just a subset of these patients that when they get an MRI and when they get the appropriate workup, they're diagnosed with this tumor. So once you're diagnosed with this tumor, of which there are approximately 22,000 cases in the US every year, usually what happens is a maximal surgical resection. So that means that they remove everything they can without damaging brain structures and get out all of the tumor from the brain. This is not a complete surgical resection because it is important to understand that GBM does invade, and so we cannot always take out every single piece of the tumor. So it's maximal. It's as much as we can possibly get out. Then you usually get a few rounds of chemotherapy and radiotherapy. And so this will be temozolomide-based chemotherapy, which we call TMZ, and radiation therapy, which is radiation plus tumor treating fields, which really directs it very specifically to the tumor and allows for higher doses of radiation. This therapy usually works quite well at the beginning, and so almost all patients will go into a complete remission. But this remission lasts an average of approximately 21 months. And at the end of that 21 months, the symptoms will often start to come back. And now this patient has a recurrent tumor. And although we may go through the cycle a few times where they have a tumor and they're able to go through temozolomide and radiation, eventually this recurrent tumor is a tumor that is highly resistant to therapy. And so giving TMZ, giving radiation has no effect on the tumor, and that tumor is ultimately fatal for the patient. So 21 months is the current average survival for a patient who is diagnosed with GBM after diagnosis. This disease carries a 100% recurrence rate and a 100% fatality rate. And that is why our research is so important. And that is why we really want to try to find ways to make this outcome better for patients. So the next logical question is, why are we getting this resistance? Why is it that when we give TMZ, even though it works at first, it eventually creates this tumor that is so therapeutically resistant? And that's an area of a lot of research, but the overarching concept there is that GBM is highly heterogeneous. And so this word has become basically equivalent to GBM, and it's one of the key things that scientists think about when they try to develop new therapies. So you can imagine that when I say GBM is a heterogeneous tumor, this is your entire solid tumor. And when you look at this from the outside, it just looks like a giant ball that looks almost like maybe it's a bunch of the same cells. But the reality is that when you go into this tumor, you will see that there are many, many different groups of different types of cells. So to simplify this, here we've just shown 4 groups. But an actual tumor will have many, many more groups of cells with different properties. And if you sort of carry this out over time, one reason that the heterogeneity leads to therapeutic resistance is that if each of these cells has different properties, each of these groups has different properties, then each of these groups might also be differentially susceptible to the drug you're giving. So in this top example, you could imagine that your blue cells are highly susceptible to TMZ, and as are your green cells. And so these just get completely killed off by your therapy. Your orange cells are somewhat susceptible, and so they get sort of cut down just a little bit. But your blue cells are not susceptible at all, and so they expand over time. And so what you're seeing here is that over time, as we track this forward, our blue cells go up and up and up because they are not susceptible to TMZ at all. And so even though we've killed off a lot of the tumor, the fact that there were a few remaining blue cells allowed this tumor to recur. And these blue cells are already not susceptible to TMZ, so we know that TMZ is not going to work, and so now this tumor is resistant. Another way to think about how this heterogeneity creates therapeutic resistance is to think about the fact that when you give a drug like TMZ, what you're actually doing is you're creating breaks in the DNA. And so these breaks might lead to novel mutations being formed, or might lead the cell to actually just change how they're expressing their DNA because they see the TMZ as a therapeutic stress. So they think of it as this is creating a toxic environment and I need to somehow make changes in my DNA to respond to that toxic environment. So whether the mutation is something induced by TMZ or whether it's something that the cell does in response to TMZ, either way you get this novel population of cells. So here these are our little purple cells. And these purple cells, since they're cells that are responding to TMZ, are again going to be resistant to TMZ. And because they're resistant, they will grow over time, and so we'll again end up with a recurrence and a resistant tumor. So it is likely some combination of both of these effects that is creating this eventually highly resistant tumor. And of course, there are way more than 4 groups involved, but this is a simple way to think about the fact that heterogeneity in GBM does create a lot of problems for developing one single therapy that can actually affect all the cells in a consistent way. To drive that point home, this is an example of a number of patient tumors that were profiled. So this is a gene expression array, which we've talked about a little bit. And so you all know that your genes are going down the side here, and your patients are going along the top here. So these are patients, these are genes. And so each line here, each column is going to show the gene expression for every gene in each patient. And while you can see that there are some overall patterns, so there's these overall patterns that we've defined as being classical, proneural, and mesenchymal, you can see that even within those, there's a lot of variation. If we look at just this box here, there's a lot of variation between each of the subtypes and also within each of the subtypes. And so while we have these broad groups that show us that there is some broad heterogeneity in GBM that can be categorized, there's also tumor level heterogeneity where every single tumor is different. The next question that sort of logically arises is how do these different groups of cells even become formed in the first tumor? How do we get all these different populations that are eventually able to give rise to this very recurrent and aggressive tumor? And so there are 2 main theories to think about that. One is clonal evolution, and the other is the concept of cancer stem cells. Both of these theories are very commonly accepted, One has not really been proved over the other, and the likelihood is that there's some element of both going on in our tumor. And so it's important to understand them both. So let's start by looking at clonal evolution. So for clonal evolution, let's say that you have a solid tumor. So this is your tumor. And within this tumor, you have a number of cells. And these cells, because they're growing aggressively in an environment that doesn't really intend for them to grow aggressively, they're constantly dealing with environmental stressors. So these are our stressors. So over time, in dealing with these stressors, these cells will sort of develop mutations. And when they develop mutations, you're going to end up with some different populations because not every cell will mutate the same. So for now, we'll keep it to 3 different populations that are kind of equal at the very beginning. So we have 3 populations in our tumor. And now this tumor is going to continue to receive stress from the environment, right? So these stressors maintain. And every cell has created some sort of mutation to try to deal with the stress. But not every cell has created an equally good mutation. And so perhaps your blue cells are not really that equipped to deal with the stressors, even though they did try to mutate. And so your blue cells will sort of disappear over time. And maybe your green cells are okay, but they're not great. So maybe a few of them will also disappear over time, but not all. And your red cells are actually doing really, really well. And they're very, very ready for this stress. They know how to deal with it. Then the red cells will sort of continue to grow over time. And so in this way, you're going to end up with a solid tumor that now has a lot of red cells, so all red cells everywhere, and perhaps a few green cells because they're doing okay as well, right? And if you expand this idea to being across, millions of cells and millions of different mutations and groups, you could imagine how you would end up with many, many groups over here, each with their own mutations and their own sensitivities and their own properties that then allow for that differential response to therapy and the recurrence. And this is basically, you're all familiar with this concept, is basically the idea of survival of the fittest, but on the level of individual cell clones. So the clones that do the best are able to survive. They're able to sort of evolve over time and make new mutations and continually deal with the stresses that are being put on the tumor. So this is one concept of how this works. Another concept of how this works is the idea of the cancer stem cell. So in this case, you will again start with a simple tumor. So this is your simple solid tumor. And within this tumor, you have all of your tumor cells that are performing all the different functions necessary for the tumor. But the difference here is that now you have a few cells that we're going to call cancer stem cells. And these are the yellow cells that we've interspersed. And these cells, just like all stem cells, so we'll take a little aside here to talk about these stem cells. So these stem cells, if we take this little window right here, this cell, this is your cancer stem cell or glioma stem cell. This has the ability to self-renew, just like any stem cell. It has the ability to differentiate, also just like any stem cell. But also in cancer, it has the special ability, because it's so plastic and it's so willing to change and differentiate and renew, and it has all of its genes kind of more open, it has this ability to also be extra resistant to therapy. And we think that is just because it is so much more able to sort of self-renew, to differentiate, so it has a lot more of that ability to sort of adapt quickly to change, and that's what makes it so resistant to therapy. So if we look at it as this cancer stem cell is very resistant and it's able to sort of self-renew and differentiate, that means that when the tumor gets stressed, so when we apply stress to the tumor, whether that's sort of stress from all the environmental stresses or whether it's TMZ stress, while all of our normal cells may die off, our cancer stem cells will remain. And an additional property of GBM is that perhaps some of our normal cells will actually turn into cancer cells. cancer stem cells. And so now we have a slightly expanded population of cancer stem cells because some of our normal cells also de-differentiated and went back to being cancer stem cells. And these cancer stem cells have all the properties we've just talked about, where they can self-renew and differentiate. And so eventually they're able to recapitulate this entire tumor. And so they differentiate to all the normal cells that the tumor needs, and they maintain a population of stem cells right here that also can still recapitulate the whole tumor all over again if necessary. And so because these cells came from cancer stem cells that are resistant, now they are also resistant. These initial cells might not have been resistant, but these new cells that came from resistant stem cells are resistant. And so now you have a tumor that has multiple different clones and multiple different groups because every stem cell differentiated differently to create a different cell, but it also has cancer stem cells that can make it resistant to therapy and make it able to regrow all over again if necessary. So one thing to point out that I think is important to realize is that this cancer stem cell theory is in a way a little bit of an extension of the clonal evolution idea, but it just focuses on one specific clone. So you can think about the cancer stem cell as being one clone in here, perhaps your red clone, and your red clone is just extra resistant and extra plastic, and it's able to handle all the environmental stressors, and so it eventually gives rise to this whole new tumor with other clones and other mutations. So one way to think about it is the cancer stem cell theory focuses on one specific clone. So CSCs is one clone. And the clonal evolution theory talks about multiple clones, so more than one clone. But they kind of all posit the same idea, that there is a group of cells that is more resistant than other groups of cells, and that is what allows this tumor to come back. And that is the same idea that we just talked about in the first slide with heterogeneity, and the idea that there are multiple groups and some groups are better than others. So that's sort of the overarching concept to understand, that you have heterogeneity, that it is led by these two things, and that is what creates a tumor that has so many different groups and can become resistant. So now that I've explained all of that, another question might be, well, how do these cells change so fast? How do they make mutations and de-differentiate and constantly adjust? And I want to bring this in now because we've talked a little bit about epigenetics and about CHIP, and so you all know the basic background to understand this concept. So we call the fact that these cells can change plasticity. The fact that they can constantly mutate, change, de-differentiate, redifferentiate, that is plasticity. And plasticity is thought to be heavily driven by epigenetics. So epigenetics is the concept that there is something else above the DNA that affects how DNA is expressed. So genetics, sort of the standard way of thinking about this, is I have a string of DNA, and now I create a mutation in this DNA. And that mutation leads to a change and a new phenotype or a new protein. Epigenetics is the concept that I have some DNA, and now some of this DNA is closed and some of it is open. And if I change modifications on the DNA, so if I add new transcription factors or new histone modifications, then I can change how the DNA is open or closed and change which genes are transcribed and thus get a new phenotype. And so when you think about the speed at which these things can happen, a genetic change is going to be relatively slow to occur because it requires a whole new cell to be formed. It requires a mutation to happen. It's a single mutation at a time. It's going to be relatively slow and low effect. Whereas epigenetics, if you change whole swaths of DNA and how they're modified and how they're opened or closed, you can very rapidly change multiple genes and how they're expressed. And so instead of having just one small change, you can now change 100 genes at once or a whole section of DNA at once. and that will create much more drastic phenotype changes. And because we see such quick changes in these cells, in GBM cells, we think that it's likely driven by epigenetic changes. So one common way to think about this that we've talked about just a little bit already is DNA that is highly open is considered usually acetylated, and DNA that is very, very tightly closed, like this, is considered methylated. And so depending on whether the cell chooses acetylation or methylation for a specific gene, that gene will now be either expressed or repressed, and that will change our phenotype. And so that's how we get these very, very quick changes, and that's how cells are able to de-differentiate so quickly and then re-differentiate again and express genes that make them more fit. And so this is an important thing to understand in the context of understanding GBM. So now that we understand some of the key background for GBM, let's talk briefly about the resources that you will almost always have available to you. So for people who are in our lab or people who want to see these resources, we will have a website where everything will be fully uploaded that you can look at. But just so you all know, a brief overview of some of the resources available would be GlioVis, which is really good for looking at expression and survival data in GBM patients. Protein Atlas, which is good for looking at cell and tissue expression, both in normal tissue and in various cancers. Stringer, which is good for looking at interactions across different proteins. And PubMed, which is of course good for reviewing all the literature that is out there. In terms of things available in your lab, you should always be aware of what protocols your lab commonly does and what they're particularly good at doing, because that will help you inform what is likely to work best for you. You will of course have whatever cells your lab has. Most GBM labs have access to both commercial and PDX lines. You will have animal models available depending on what your lab does. And there may be existing data sets in your lab already that look at the question you're interested in, and that might provide a good point of reference or corollary to your data. Another thing to keep in mind is that now that you have the background and you kind of understand the language of how to talk about GBM, the burden is really on you to evaluate ideas and think for yourself about next steps. There are a lot of different ways to do things and think about things in science and in research, and everybody sort of has their own way, and so you should really try to understand everyone's way, but make your own decisions about what you think makes sense. So we've covered the basics of GBM, and that was really intended to just give a refresher on the background that we need to think about a good scientific question. So now let's move forward and talk about how to actually develop a hypothesis. So we'll start with the formal scientific method. And I'm sure this is something everybody has seen since middle school onwards. But although it is a very commonly thrown around diagram, it can be a helpful framework to think about how to develop a question, a hypothesis, and then how to test it. And so the basic idea is that you would ask a question or find a problem that you're interested in. You would do some background research on that problem, and that would allow you to generate a hypothesis about what the answer to the problem or question is. So your hypothesis is your question plus your answer. You would then design an experiment that tests your hypothesis, so this needs to be testable. And you would analyze that experiment and make some sort of conclusion. This conclusion might be that my experiment didn't work or didn't give me an expected result. And so then you would start all over again with, okay, now I have this new research and this new question. Let me generate a refined or a new hypothesis and do this all over again. Now, this is a very formalized way to think about it, and it has been criticized for not being plastic enough or flexible enough to really inform scientific discovery. And so another way to think about it would be to think more about well, how do I actually get my question? My question doesn't just come from nowhere, right? It's not like I'm sitting around and one day I'm like, I have this great scientific question. It has to come from something. And so a better way to think about your scientific question and your scientific method might be to think about it as being informed by things you know. So now that we know all this GBM background, you might have questions that are informed by knowing all that background. And so you can think about it more as, oh, I noticed this thing that was kind of interesting in the background we learned. Or I wonder if this other thing that I learned about in a different class happens in GBM. Or maybe it's that I did some experiment and my experiment just completely did not work. And all of these things together, plus any other observations you make or things you learn about from papers, from classes, from your own research, all of those can lead to a question that you think is interesting and a hypothesis that you would like to test. So a more plastic and simple way to think about it is anything that you think is weird or interesting or notable can easily be turned into a question, a hypothesis. So to expand on that a little, how do you choose a research question? Because your first step in developing a hypothesis is identifying a good research question. So we talked already about how you might get that from something you notice, but turning it from something you noticed into an actual question can still be a bit of a process. And so part of that is, again, going back to understanding your background. So really the process looks more like I noticed something interesting, and now I'm going to read about it and do some research, and then I'm going to come up with a question, and then I'm going to do some more research to figure out what the answer might be, and then I'm going to come up with a hypothesis. So right now we're sort of in this phase of I noticed something interesting, and I want to research it and come up with a question. And this research part is really, really important because it tells you whether your question is good and whether it makes sense, whether your question has any merits. If your question is inapplicable, if what you're asking about just there's no evidence that it occurs or that it is even interesting or there's evidence to the contrary, then perhaps it's not a great research question. So your research question can really be anything, but it's important that it be logical based on the background. Now, I'm not saying that if there's nothing out there, you shouldn't research it. That's a novel concept that maybe you should go after. But if there's things that are directly contradictory, then that's where you might want to start and rethink your question. So, places your questions might come from. I'll start with large screens because we've talked a lot about large screens already. And so these screens can lend themselves very well to a very broad question about what that screen might show. So you could say something like, I wonder if I screen all the genes in the genome for which ones are important in driving viability of GBM, what I would find. And that's a very broad question because you don't know what you will find, but you do know that you will find something if you screen across the whole genome. So that's why I say it's a broad research question, but a very useful one that you can then filter down. And so another question that might come from the screen is that I actually did the screen and I found these 10 genes that were interesting. And now I want to ask a question about why these genes are important or what they're doing. And so that's another research question that can easily come out of a large screen. A second thing is looking at literature or looking at other things you've read about, learned about, or heard presented. It may be that something in that piques your interest or there's something very interesting or topical. For now, that topical thing would probably be the coronavirus that's going around. And so that might be something that you want to look at or ask a question about. And then finally, if you're in a lab, there may be ongoing projects that would be a good place to start. So perhaps there's a tissue bank that you have that you can easily ask a question about. Maybe there's a study where you got a strange result or an unexpected side finding that you want to investigate more. And so those are also good places to find research questions. So now that we have a research question, how do you figure out what background you need to make sure that it is actually a good question? And so for GBM, we've discussed a lot of the basic background you would need. But in general, you should think about what experiments you might need, what genes are involved, what diseases are involved, and sort of research around those questions. So do you have the experiment? Do you know how to do the experiment? Can you get the resources to do the experiment? Are there genes that do specific things that are involved? Like maybe you think that a gene is involved in mitosis, but when you research about it, it's actually really important for protein translation. So then maybe that gene really isn't involved in mitosis because it can't be, because it's involved in protein translation. And then you would know that maybe that's not the best question for me to pursue. So this background part is hard to explain until you actually have to do it, but it's very important to take a question and really break it down into all its subcomponents and make sure those components line up and make sense, because if they don't, then your question probably isn't going to work out for you. So let's just go through two examples. So one example would be, you might ask, what are the expression patterns of ACE2, which is a gene, in humans, and how might that influence which organs COVID affects? So a presupposition of this question is that ACE2 means something about which patients COVID infects or which organs COVID infects. Because I'm asking, what are the expression patterns of ACE2 and how does that affect the organs that COVID affects? So I'm saying that, like, I think that ACE2 must be involved with COVID somehow. And so this is the background question I would want to ask, is there some link between ACE2 and COVID entry into cells? And as it turns out, there is quite a big link. It's been very definitively shown that ACE2 is one of the major receptors for COVID entry. And so because this holds true, I can then go ahead and investigate my question of, well, what are the expression patterns of ACE2, and draw a conclusion about, well, ACE2 is really high in the lungs, so maybe COVID affects the lungs very strongly. Another question you might ask for background here is, do I even have a way of checking ACE2 expression levels? So do I have the tissues? Do I have the cells? Do I have the things I need to actually check ACE2 expression across all the organs in humans? And this is more of a materials and resources question. If you don't have the materials or resources and you have no way of getting them, then perhaps this question is a bust because you're just not able to do it. And so those are two simple examples of background questions you might look at. A similar variation on this question, we might ask, well, how does ACE2 expression in the brain lead to SARS CNS infection? So SARS is a similar virus to COVID. And we've already talked about how the first question here, the first assumption in this question, is that ACE2 and SARS infection have something to do with each other. And this checks out because SARS is similar to COVID. A lot of the research around ACE2 was done when SARS was a big deal. And so we know that SARS does need ACE2 to get into cells. So this checks. Another thing that this question sort of assumes is that SARS can somehow cause a CNS infection. And this is unclear. So perhaps it does, but there's no real clinical data that says that SARS directly goes in and creates neurological symptoms. It's mostly a disease of the lungs, just like COVID is. And so it would be hard to draw a conclusion about how ACE2 causes CNS infections when SARS doesn't actually cause CNS infections, right? Your presupposition in this question is incorrect, and so this question doesn't really hold any merit. And so when I talk about finding the appropriate background, I really mean you need to make sure that all the assumptions in your question hold true, that your question actually makes sense and everything in it makes sense, and then you can actually go ahead and investigate it. Okay, so now that we understand sort of how to make our question, let's talk about how to develop a hypothesis for that question. So a simple way to think about a hypothesis is to think about it as a statement that encompasses your research question and your proposed answer. So a simple declarative statement that is informed by observations and background, as we've talked about. You need to make sure the background lines up. But essentially, assuming the background lines up, it's going to be a simple statement that says, this is my question and this is the answer. You can think of it as a simple if-then statement. So if a certain condition, so if condition then proposed answer. This is what you think will happen in this condition. This is the simplest form of a hypothesis. And this is very testable because then you would just expose to condition and look for what you think is going to happen. So some simple examples of this could be, maybe your question is, what happens if I knock down p53 in cells? And your answer is the cells should proliferate more. Then your hypothesis would be something like p53 knockdown results in increased cell proliferation. Or as an if-then statement, if I knock down p53, I will get increased cell proliferation. That's a simple hypothesis. You can also go the other way and say, well, my hypothesis is that p53 overexpression leads to reduced proliferation. And then if we broke this into a question and answer, the question would be, what is p53 overexpression doing? And the answer would be decreasing proliferation. So you have to have the skill of taking your question and turning it into a hypothesis. And really the important thing there is that you have a proposed answer. You have something that you think it's going to do. So that was a very simple hypothesis. But hypotheses like that are always getting refined because it's too simple, it's too basic. And so we want to make something that's a little more refined and a little more elegant. And so perhaps for a more refined hypothesis, we would want to include the question, like we've talked about. But we'd also want to include any relevant details in that question. So maybe we want a very specific type of p53 mutation, or a very specific change, or a very specific cell type, a specific experiment. So this could be anything. It could be experiments, cells, tissues, specific modifications. It really can be anything. But some sort of relevant detail that sort of focuses your question a little bit more, instead of being like, well, in every cell in the universe, what does p53 do? Maybe you would say, in this specific group of cells, what does p53 do? And then for the answer piece, you would want to give, well, I think p53 increases proliferation or decreases proliferation. But you would also maybe want to say a little bit about why that matters to you, right? Because why should someone care about your hypothesis? It's important to tell them why they should care. And so the significance of this would be that it has a role in cancer. And so you might want to mention that in your hypothesis. And again, this will be phrased as a simple, declarative, very, very clear statement that is clearly testable. So let's look at some examples of this. So here now we have a question that says, what happens if I mutate p53 specifically by changing the fourth amino acid? So you're making a very specific mutation here. You're not talking about every single mutation under the planet of p53. And your answer might be that p53 gets inactivated and the cells proliferate. And the significance of that is that it creates increased growth and aggression of cancers. So this hypothesis might sound something like, I will mutate p53 by changing the fourth amino acid, and that will result in inactivation of p53, increased cell proliferation, and therefore increased aggressiveness of my cancer. So that is, again, a simple and straightforward statement, but it is a lot more involved than the first hypothesis we created. And it's a lot more specific, it's a lot clearer, and it tells the person why they should care about my hypothesis. So let's say it again. So you're going to say, if I mutate p53, then I get inactivation, and then I get proliferation, and then you explain why this should matter to the person. So you're being very, very clear about your hypothesis. Another example of a hypothesis would be something around a large-scale screen. So here we say, examining RNA-seq data, which is our screen, so this is our detail, before and after TMZ therapy. So our question is, what is TMZ therapy doing? And then you say, it will allow me to identify genes that are elevated after therapy. So this is your answer. This is what the screen will give you after you ask your question. And then you say, these genes may be involved in TMZ resistance. And so this is your significance. It's telling the person, why should they care about these genes that you're going to find. So this is an example of a very, very clear and refined hypothesis. And you can see how it's better than before because we have all the necessary details. We've sort of focused our question down to one specific scenario, so that makes it much more testable. And we've told the person why they should care about this hypothesis and why it's important in science to do it. So I would encourage you to make hypotheses that look more like these examples. But a good way to start is to think about the simple if-then statement or the question and the answer, and then think about, well, what details can I add to refine this down to one simple scenario, and what significance can I add to make a person really care about this? Now that we've talked about how to create a good hypothesis, let's talk about how to design that experiment to validate your hypothesis. So let's just start with some basic principles that should go into your validation design. The first thing is to keep it as simple as possible. The first experiment to validate your hypothesis should be the simplest and most direct way to check if your hypothesis is true or not. For all questions, a good way to do this is to simply boil it down to the most basic elements and then construct, moving forward from there. So, for example, if we were talking about p53 and saying a mutation in it will lead to inactivation and increased proliferation, the really simple experiment would be to mutate p53 and check for proliferation. And an even simpler way to just know if p53 is even involved in proliferation is to just look at p53 and proliferation. So you want to make it as simple as possible so that it's like a very executable experiment and something you can quickly check before you move forward and devote a lot of time and resources. The second thing is to be aware of what you have. So there are a lot of experiments out there. You won't have learned about all of them. There's too many for anyone to learn about all of them. And so sometimes you have to go look for the appropriate experiment. So, for example, above, you might not know the right experiment to look for proliferation, but there are definitely assays that allow you to look for proliferation, and you need to go find the experiment that allows you to do that and make sure it's doable and something your lab can have the resources to do. You also should be aware of what your lab has. So sometimes the lab might already have protocols in place to look at certain things, and so that's also a good way to know what experiments you can do easily to validate your hypothesis. And then finally, the most important thing for any validation experiment is to make sure that you know your controls. And that is what allows you to know whether your experiment worked or not. And so you have to use positive and negative controls, and you have to make sure that the assay you're using, if you got an assay, that the assay itself is actually working the way you expect. And that's what the positive and negative controls will allow you to see. So with those principles in mind, one of the things we talked about was making sure that you know what experiments are available to you. So let's take a quick aside to talk about what experiments can you do. So all the screens that we've learned about are probably available to you. So for example, we talked about using CRISPR-Cas9 for gene functionality, DROP-seq for single-cell mRNA, RNA-seq for global mRNA. We talked about mass spec for protein expression, and then we talked about the IP experiments that allow you to see interactions between proteins and other proteins, and also between proteins and DNA. So all of these screens are available to you, and these might be a good first step for a very, very broad hypothesis that specifically implicates these screens. But in general, these are not your first step for validation. If you have a simple hypothesis, your first step should not be to run to a giant screen. These take a lot of time, a lot of energy, and a lot of money. And unless the hypothesis specifically points to them or specifically says that this is the best way to do this, this is probably not going to be your first step. But you should know that all of these are available as your hypothesis develops, and if they become necessary or seem like the best step, they're definitely a good idea to do and can lend a lot of power to your analysis. So the more reasonable and more common first steps for a basic hypothesis are your molecular biology techniques or whatever types of techniques your lab routinely uses. That's usually where you want to start. And there are always new assays being developed, and so this is kind of where it comes in of like looking for the appropriate experiments for what you want to do. So, like I said, these usually are your first step for a simple validation. So let's talk about these step by step. So the first thing we're going to talk about is Western blots. So these are for looking at the levels of a single protein in a certain condition. And you always want to make sure you include the appropriate controls with them. You can do qPCR, which similarly will allow you to see levels of mRNA for a single gene in a condition. And again, controls are very, very important here. MTTs allow you to see viability of cells and proliferation of cells. Your in vivo studies are a little bit more involved, and again, generally not the first step, but they are a good second or third step for validating a hypothesis. And these will allow you to see animal survival and tumor progression. You can also do IP Western blots, and these will allow you to identify protein-protein interactions, but you have to remember that these are going to be for a single protein of interest. So unlike the large IP mass spec we did, where you could identify every protein binding to a protein of interest, these will be protein of interest and will allow you to identify maybe one or two proteins that are binding to that protein. And then finally, we have immunohistochemistry, immunocytochemistry, immunofluorescence. All of these terms mean something slightly different. So this one refers to tissues, this refers to cells, and this refers to technique in general. But the idea is that all three allow you to see specifically where proteins are localizing in cells and tissues and whether they're localizing with each other or not. So it's very powerful for seeing where proteins are in space. So these are kind of your basic molecular biology techniques. Like I've mentioned, there are many, many more that you could use. And there are things that are being constantly developed that are new. But this is probably where you'll start, and we'll talk more in detail about all of these different techniques in future videos. And then I just want to briefly touch on what tools you have to make modifications and to study these things. And so one of the tools that is available to you is sort of your models. And so here you can think about any sort of cell line that you're interested in, tissues, and animal models. And then in terms of looking at function of certain genes, it can be very helpful to knock down or overexpress those genes in order to tie it to a specific effect. And so we have plasmids, they can be CRISPR plasmids or shRNA plasmids or reporter plasmids, and these plasmids will allow us to make modifications to cells as desired. So remember that you cannot tie expression of a gene to a function unless you've shown directly that knocking out that gene or overexpressing that gene results in a change to the function. So that's why these are so helpful, because they allow us to do knockdowns and overexpressions. And then reporters specifically allow us to have a measured output of what the gene expression looks like. So for example, you can tie notch expression to GFP and then just measure GFP, and by that get a quantitative view of how much notch is changing. So these are really important tools for just seeing gene function and how it changes in cells. So now that we have the basic tools and we kind of have some framework for how to think about validation, let's talk about some examples. So we'll start with our very simple example of knocking down p53 should result in increased cell proliferation. So how would you validate this? Remember, we're going to boil it down to its simplest elements. So we want a knockdown of p53 and we want to measure cell proliferation. So we would probably take one cell line to start with. We would knock down p53 in that cell line, and then we would take control and knockdown and compare proliferation, likely using an MTT assay, because we've talked about that and we know we have it in the lab. So that would be a very simple experiment to check this hypothesis. And then, of course, if this experiment worked, you might ask other questions about, well, what specific kind of modification results in a p53 knockdown, or how much does cell proliferation really change? et cetera, et cetera. So there's many ways to sort of refine this hypothesis and ask additional questions, but this would be where we'd start. Another example of a hypothesis validation would be this one. That was the more complex hypothesis that we talked about as an example. So we talked about this hypothesis where we said examining RNA-seq data before and after TMZ would allow identification of genes that are elevated after therapy and may be involved in resistance. And so this is much, much more involved, but this hypothesis really points you very directly to the experiment you're going to do. So in this case, you would just take your RNA-seq data and you would take an RNA-seq experiment and set it up. And so you would do before and after TMZ, so no TMZ and with TMZ. And you would compare the results and see what genes come up. And that would be your simple validation. And in this case, it's okay to start with RNA-seq because your hypothesis really points you very directly to RNA-seq. But in this case, you would not want to start with a giant CRISPR screen or RNA-seq screen because there's a very simple experiment that you can do first to check whether this is even correct or not. So the final point I want to make about hypothesis development is that so far we've been talking about one hypothesis and one experiment to validate it. But as I'm showing you in these figures from a paper in Cell Reports, you can see that these are multiple experiments. And these experiments and these kinds of papers don't come from having just one hypothesis and one validation experiment. It's usually a far more complex structure than that. So I want to start getting into a little bit of how do you organize a project like this and how do you understand the different levels of hypotheses that go into a project like this. So here's a basic framework for how to think about multiple hypothesis levels. So at the top, you have your sort of overarching project definition, so your overarching question and your major hypothesis. And a key point is that as you move through your multiple sub-questions and experiments, this hypothesis will likely get refined and changed. So you might start with something very simple, like what we started with as our simple hypothesis example. And over time, you will refine that hypothesis to be clearer and clearer. And so eventually you have a hypothesis that really encompasses all of the work you did over here. So we'll take this overarching question and hypothesis and we'll divide it into 3 logical sub-pieces. And each of those pieces will have their own question they're addressing and their own hypothesis. So for example, if your overarching question was looking at expression of genes before and after TMZ, then your aims might be doing a large screen here, sort of validating some of the targets from that screen, and then identifying mechanism for one of those targets, or something like that. So it needs to be 3 very logical breakdowns of what the initial question was. These ideally should not bring in extra outside adjacent information. It should be very focused from the question to the sub-question. And then based on your sub-questions or aims, you will decide specific experiments. So you might say, for my screen, I need to do my screen, and then I also need to find a way to validate that screen, and I also need to find a way to filter down my targets. And then once you've filtered your targets, you could move to the next step, which will be actually validating those targets. And then you'll define a series of experiments to validate those targets. And for any of these experiments or questions, it might actually break down into even more experiments. And so you do want to keep that in mind. This is not the end. You might have further divisions even after this. And so this is just a framework for understanding how do we build up to those really big projects and really big papers. And the idea is basically that you have a hypothesis at every level and a question you're testing at every level until you get down to a single experiment. And then you sort of build it back up again. So let's do an example with just 2 levels. And so here we'll say that our question is, how does TMZ affect GBM levels? Now that's a pretty vague question, so let's frame it as a hypothesis. So our hypothesis might be that TMZ treatment results in reduced proliferation or reduced viability of GBM cells. That's a very, very simple hypothesis that comes straight from this question. So we're not expanding the question or changing it. We're just taking a very simple hypothesis from it. An example of a more complex hypothesis would be to say something like using RNA-seq to really understand global gene expression changes. And that's also a good example, but that's sort of broadening this question and making it a very big project. So for now, we're just going to focus on this simple and straightforward question of TMZ treatment affects cell viability. So if we were breaking this down into sub-questions, some logical sub-questions you might ask if I told you TMZ treatment affects cell viability might be, well, at what dose? How much TMZ do I have to add before it's going to affect viability? Or you might ask, well, how much time will it take? Is it an immediate effect? Does it happen in like 10 minutes? Or does it take 3 or 4 days? Or does it take even longer? And a third question you might ask might be, well, what types of cells? Is it a subtype specific effect? Because we talked about how there are multiple different subtypes and some are more susceptible to TMZ and some are not. So you might ask, well, which cells are killed off most effectively by TMZ? So those are all logical sub-questions. What would not be a logical sub-question of this question that you've asked is to introduce a new gene or a new concept, because those are not part of the initial question you stated. For example, if I were to say, well, what is the effect of P53 on cell viability when I treat with TMZ? That is a question of how TMZ affects GBM cells, but it's a question that now is bringing in P53 and a number of other genes, which is not the question that we had initially asked. The question we had initially asked is just a simple, how does TMZ affect GBM cells? So you want to try to keep it very straightforward so that you don't start bringing in a lot of adjacent things. You might decide that you are actually interested in P53, and then maybe this question changes, and then it would be appropriate to focus this question on P53 and change your experiments accordingly. But for the question we've asked, we really want to keep it to the question that we have asked and make a logical next step from it. So let's just do one of these as an example. So let's say that I said my overarching question was, how does TMZ treatment affect viability? And my sub-question for that is, specifically at what dose of TMZ treatment does cell viability get affected? So the hypothesis for this might be something vague to start with because I don't really know what the answer is. The hypothesis just might be, well, as the dose goes up, I expect that the viability will go down. And the experiment I would do would be to do an MTT and look at multiple doses, so maybe like 0, 2, 4, 6, and so on, up to like 50 micromolar, and look at viability of cells across all these different doses. And of course, this would function as my negative control. And perhaps I could add a very, very high dose, like 1,000 micromolar as a positive control to make sure that all my cells die here and none of my cells die here. So these are ways to sort of test the hypothesis of what dose. You could similarly test how much time by varying times rather than doses. You could also similarly test what cells are affected by varying the cell type rather than the time or the dose. So this is a very simple example of how we might break down a question and set up one experiment to answer each sub-question. So having done that simple example and walking through a few examples of how we do these things, let's go through one large project example. So this is going to be an example of a large-scale screen. And as a reminder, this is the pipeline that we had initially talked about for a large-scale screen. So for a screen, you would ask a very broad question to begin with, and that question might encompass the whole genome or a very, very large subset of genes in the genome. You would then interrogate appropriately, whether it's a CRISPR screen or an RNA-seq or whatever type of screen, across the genome or proteome. And from that, you would be able to identify significant targets and pathways that matter. And so this is all still just large screen. And then when you cross this line, you've used other data sets and other sources and more information about the genes to choose maybe 1 to 10 genes that you're actually interested in. So this is where you move into your validation phase, where you're like, I have genes that I'm interested in, and now I want to know if they actually work. And then finally, with maybe just 1 or 2 genes, you might choose to identify a mechanism. And all very strong papers do try to identify a mechanism for how these genes function. So 1 to 10 genes you might validate and say, yes, these genes are involved in whatever I'm talking about. But that still doesn't explain how they're involved. And so this final piece is identifying how they're involved and what they're doing. So let's say our question is, what genes and pathways confer susceptibility to TMZ in GBM cells and how? This is our first research question. This is the question that we're going to be using moving forward for this example. So the first thing you should ask yourself is, what background do I need to answer to be able to answer this question? And for this, it's really a lot of the background that we've already talked about. You would want to know, what is the link between TMZ and GBM? Why is it important whether cells are TMZ susceptible or not? What does that mean for patients? What kinds of screens are out there that might allow me to do this type of experiment or test this kind of question? And maybe you would want to know a little bit more about the outcomes for GBM, right? Because that would sort of inform why this is important. So those are all things that would be useful to know. And if you evaluated all those questions, you would see that there is a link here and here, that there are screens and that there are bad outcomes. And so all of that would lead you to decide that this is probably a good scientific question that is worth pursuing. So now that we've decided it's a good question and we've established our question, we are going to say that we know the background and we know it's a good question. So now let's move on to establishing a hypothesis. So our question was essentially, which genes lead to TMZ susceptibility and how? And if we said this is a hypothesis, we might say something like, using a CRISPR screen, because that is, of course, the best technology to identify this type of thing. Using a CRISPR screen, we will identify genes and pathways involved in susceptibility. That might be our starting hypothesis. We can't really say more than that just yet because we don't know what kinds of genes and pathways we're going to get, but this is something we will continue to refine as we find out more and more. So if this is our overarching hypothesis, we now need to divide this into 3 logical pieces. So the first piece will certainly be to do the screen. And for this, the question and hypothesis stay kind of the same as what we've already talked about here. Because we've already said that we're going to use a screen in order to identify genes and pathways involved in susceptibility. So that basically covers our question or hypothesis for this screen. The second logical thing would be to take the genes from this screen, because we also want to know how these genes are involved in the screen, which means we need to choose some genes to investigate further. So the next thing would be to choose some genes and validate them at the bench, right? Make sure they actually work in real life in multiple conditions, not just in one screen. So now we choose our genes and we validate. But this still doesn't answer the question of how. This will simply answer the question of, these are genes that are truly important for TMZ susceptibility, but we haven't gotten to the how yet. So the final piece of this will be to identify the mechanism, which is the how. And this kind of together will answer our overarching question, which is, what are the genes and pathways involved and how do they work? We've kind of broken that down into 3 major steps. And you can imagine that as we move through these steps, we will be able to update our hypothesis as well. So this is what we just discussed. We identified a hypothesis that will need to be refined, and we identified the 3 pieces. And now we're going to go forward and identify the specific experiments or steps that need to be taken to accomplish each aim. So for doing the screen, we need to perform the screen. We will then need to analyze the screen. And we'll then need to do some basic validation and quality control of our screen results to make sure our screen actually worked. So this is not validation of specific targets. This is just validation of your overall screen. Is my screen logical? Did my screen work? Are there any major issues in my screen to be aware of? This is not the point where you pick specific targets and validate them. So for now, we're just going to focus on these three things. So the first thing to figure out is, if I'm going to do these experiments, how am I going to set them up? And we have already talked a little bit about how to set up a simple TMZ screen. So this is just a reminder. This is from a paper in Cell Reports that did this screen. And they used what we've talked about in the past, a genome-wide sgRNA library and an immortalized GBM cell line. They chose to use a control with a low dose of TMZ compared to a high dose of TMZ, but you can certainly do this with a control and TMZ or with some sort of other condition. And from this, they were able to identify certain genes involved in resistance and certain genes involved in sensitivity. So this was their overall setup. And once they actually performed the screen, the next piece, as we talked about, was to analyze the screen. So in their analysis for sensitivity, this is their plot showing that there were 4 genes that really came to the top. And this is for resistance. And this is showing, again, that there were a few genes that really came to the top for them. And so this, again, sort of seems logical. There are other papers that have identified these genes as being important for sensitivity and for resistance. And so for a basic analysis, we have our basic analysis, and we know which genes have come to the top for significance and fold change. So we've done our second piece of our project. So now we'll move to the third piece, which is simply understanding whether the screen itself worked or not. And one way to do this is simply to put all of your top targets into something like Strinker and see what the interactions are. So for this, you can see that they found a number of different mismatch repair pathways or DNA damage repair pathways. And that is logical with what other screens have shown for TMZ susceptibility genes, that it tends to be a number of mismatch repair pathways. Other things you could do at this point would be to look at the genes themselves that you found and validate by looking at other similar screens and see if they found similar things. You could also try things like looking at the screen quality and making sure there are no quality issues, and looking at your overall gene distributions and seeing if there are any notable patterns. All of these things will tell you that your screen worked. So now that we've sort of done our screen and analyzed it and validated it, we know that we have a reasonable screen that we can move forward with in order to actually choose targets of interest and move forward. So we've done this, perform screen, and we've done these things. And we've been able to further refine our hypothesis because now we're able to say that, we know that mismatch repair is important. So now we can add that we want to understand how mismatch repair functions to promote susceptibility, right? And that kind of addresses the how piece, which genes and how do they work. So we're able to refine our hypothesis and we're able to move forward with our next steps. And our next steps are going to be to really validate the targets. So now we're at the point where we have chosen something that we're interested in. We have a screen. We're able to choose targets we're interested in. We're able to validate those targets with other potential resources. And then we're also able to validate at the bench. And this will turn into a number of other experiments. So ways you could validate this at the bench would include looking at viability with MTTs or looking at survival studies with mice. And so this will create 2 more branches with 2 more experiments and 2 more hypotheses that need to be done. So first, let's look at target choice. So our first aim was all about doing the screen. And we did the screen and we checked that it was fine. Now we're on our second aim, which is where we really need to choose our targets of interest and validate them. So we've talked about this before, but as a reminder, some factors that might go into choosing your target are obviously the results of the screen. So which genes are most significantly changed, or which pathways are most significantly changed. You might also, when choosing specific genes, care that they come from a pathway that's very enriched. Or you might care that they come from pathways that you are able to study in your lab. And then finally, it might be good to look for things that are highly novel so that you're not scooped by some other group and you can actually produce something with enough time. And then finally, if you do have other data sets in the lab or you have access to computational data sets online, it might be worth looking at how your genes perform in other data sets. So again, as a reminder, these are their figures, and you can see that mismatch repair is a big pathway that's important, and these 4 genes are their most significant genes. So looking at just this, knowing that these 4 genes are their most significant genes and that they're involved in MMR, and that MMR is highly enriched, you might say that it seems very logical to choose these 4 genes as the genes to move forward with. So for now, we'll just say that we chose these 4 genes and we want to look at them even more. So for looking at them even more, a quick way, the paper did not do this, but a quick way to look at whether these genes are important or not in GBM is to look at them in data sets that we have available to us. So for example, MLH1 is what we're looking at here. We can look at GlioVis expression data, and we can check how these genes change after TMZ exposure, because this is increasing TMZ exposure. When you go from primary to recurrent tumors, that's a patient that has received TMZ. And so you can look at how MLH expression is changing. And you can see here that it's not changing very significantly between primary and recurrent, but as you get increased recurrences, you do see increased change and increase in MLH1, which suggests that it might be important. You also see that there are survival changes, and you can see that when you look at normal versus GBM, there is a significant increase of MLH1 in GBM cells as compared to normal. But there are some key points to make here. It's important to look at the right data set for what you're interested in. And out of these 3, the most interesting one is definitely this one, because this is a direct look at TMZ and how it affects MLH1. These patients are getting increased amounts of TMZ. In this case, we have primary data and recurrent data, but we already know that MLH1 is not really changing significantly between primary and recurrent. So this is not very helpful to us. And then this compares normal versus GBM. And while it's interesting to know that MLH1 is elevated in GBM as compared to normal, our experiment is about whether MLH1 promotes TMZ susceptibility. So what we would really want to look at is GBM versus GBM treated with TMZ, because that is the condition we care about. Nothing in our experiment really looks at normal cells. So this is less useful to us because it's comparing 2 conditions that are not very relevant to our experimental condition. Our experimental condition was looking at which genes promote TMZ susceptibility. So if GBM plus TMZ had even more expression of MLH1, and that expression of MLH1 was associated with longer survival, then that would be interesting. But normal versus GBM is not telling us very much about the question we're actually asking. So while these resources are very, very useful, it's important to look at the right graphs to answer the question you're actually asking. And in this case, I would say this graph here is the one that's the most applicable and the one that's the most interesting. And then the final piece would be to validate at the bench. And so here, this is an example of an MTT assay. And you would expect that if MLH1 promotes TMZ susceptibility, the cells that have MLH1 would die. So if we knock out MLH1, so this is now MLH knocked out, we would expect to see no cell death with TMZ addition. And when we look at these knockouts, these are knockouts of MLH1, you can see that the red and the yellow stay pretty stable even after increasing doses of TMZ, which tells us that these genes are actually involved in cell death. Another thing you could do is test mouse survival in vivo, and that would also help you understand whether MLH1 plays an important role in TMZ susceptibility. And then finally, now that we have validated at the bench and chosen our targets and completed our screen, we can move on to choosing a target to identify a mechanism for. So for this case, let's pretend that we chose MLH1 to identify a mechanism for. And we can go ahead and further refine our hypothesis and say we're going to specifically focus on how MLH1 plays a role in mismatch repair in TMZ susceptibility. So mechanism development is fairly difficult and it requires a lot of iterating. So today we're just going to discuss some basic aspects that might go into it. So when you develop a mechanism, some things to think about and the way to approach it would involve first reading as much as you can about the gene. You really want to know everything that is known about the gene for which you want to identify a mechanism. You should look for how it interacts with other genes. So stringer might be useful here. You should look at what is known about it. And then you should also look at other papers that are published that might propose a mechanism that you could reapply to your specific scenario or that you could adapt or build on. This is how you build on other studies. And this is the best way to sort of start thinking about a mechanism. It's also why it's useful to choose a gene that has been somewhat studied so that you can have something to build upon. The second thing to keep in mind is that you might go through a few iterations. So you construct a hypothesis based on your reading, and then you test it, and then you construct again based on what you get. So this is one of those situations where your experiment is going to have a lot of branches, it's going to take a lot of tries, and you might need to go down a few routes to find something that works. And then finally, this is always important, but especially important here, make sure to plan your experiments very carefully. So plan the most specific and simple experiment you can to test the area of interest. Try to avoid any detractors, make sure your controls are really, really good, and make sure that you find the appropriate assay. You might need to go looking for a new assay to find the thing you need, but go looking for the assay that really answers your question directly. So as a brief example, let's say that we looked at MLH1 and we know that it's involved in DNA damage repair. And we also know that TMZ creates these double-stranded breaks in DNA that you see here, and that the DNA damage repair system, or the MMR system, is what comes in and repairs these. And now we know that MLH1 plays a role in MMR. So you could put these facts together and try to generate something. For example, you might say that I know that MLH1 plays a role in MMR somehow. And I know that having TMZ creates breaks, and MMR helps cells resist TMZ by going and repairing those breaks. So maybe there's something about resistance here. And so when I give TMZ, the breaks are created and MMR is activated. And so this creates activation of MMR. And perhaps MLH1 is what actually allows MMR to get activated, or perhaps MLH1 is what causes MMR to get deactivated. And that's what creates the sort of mechanism of MLH1 being involved in mismatch repair and then leading to susceptibility. So that was a little complicated, and you can break it down for yourself step by step. But the point is to try to string together the few facts you know in a logical mechanism and then test that mechanism. So what we just said was something like MLH1 has some role in DNA damage repair. That was your first statement in this mechanism. And so that's the first thing you should test. So if you want to look at whether MLH1 has a role in DNA damage repair, you need an assay that measures DNA damage repair. And this is an example of a place where you should probably go look for the right assay that really directly measures DNA damage repair in order to see what is going on with MLH1. So the assay here is PH2AX, is a way of looking at DNA damage. And doing this assay, it's a simple antibody stain, is a really direct way to measure what you're interested in. So you would have had to go find this assay. It's not something we talked about, but it's a very simple way to do it. And so that ties into finding the right thing to do. So if you did this assay with and without MLH1 and you confirm that MLH1 has a role in DNA damage repair, then you might move on and say, well, the next piece was to say that TMZ somehow affects this process. And MLH1 has a role with TMZ of changing the DNA damage repair response. So now we need to add in TMZ and look at MLH1 control and knockdown and see how that affects the process. And then finally, we might take whatever mechanism we found once we've iterated on this a few times and found a mechanism we're happy with. We might take that and test it in animals because, of course, we want to make sure that it really does carry over to humans and really is happening. So we might go and test with the knockdown and with TMZ in animals. What do we see? So that was just a very simple example. Usually it takes a few different takes to figure out the mechanism, and it might involve a few different links. This was sort of one simple link, MLH, MMR, TMZ. But it might take more than that to really figure out what is going on in this mechanism. So we'll practice this more in upcoming lectures, but this is one of those skills that also just takes time to develop. So I would try to apply it to any experiment you're doing in the lab or try to pick a gene and just practice. And that way you'll sort of develop that skill of being able to do it on the spot. So to review, today we talked about understanding a research question, developing a hypothesis, how to break down a project, and a little bit about how to think about mechanisms and validation. If you have any further questions, you can feel free to contact us through any of these methods. Thank you so much for listening.